Perfect. Maybe a little loud. Do you want to mic test that?
Next month, we'll all be in person.
God, I hope so. Oh, do I need to mute the owl? Is that the thing? Or are we good?
No, you're good.
Okay. All right. So at 7:01, we're going to get started and kick off for this evening. Thanks to those of you who came in person tonight, and welcome, of course, in terms of our online audience. I'm sure people will be joining. It usually takes a few minutes for things to come together. Personally, I truly can't believe that it's September this year. That's been quite a ride. But I'm going to share a little bit about things that have been exciting for the center and just give a little bit of background before I introduce our speakers. First of all, of course, welcome again, and a huge thank you to UCI Media Services, to our communication folks, Judy Beck and Abby Enriquez from the center, to Brian Cummings, who is always really critical in terms of our organization. And I thought I would just pause for a minute because we're starting out a new quarter at UCI and call out a few things, both about the campus and about the Stem Cell Center over the last few years, the last few months. It does feel like years though. It just came out and it was announced this week. So this is the Points of Pride background gift that you can get from UCI Media Services, just things that are going right here. And the exciting bit was both in the US News and World Report college rankings that just came out, and in the money rankings that just came out. UCI actually moved up multiple slots, so I think we're now listed at number 8 and number 9 respectively in terms of public university rankings across the US at the national level. Hugely exciting, and of course we've retained this slot that's really unique to us in terms of diversity, equity, and inclusion for public universities that do the most to encourage upward mobility, and so we're really proud of that here. The Stem Cell Center, as most of you know, is composed of a really diverse set of faculty ourselves, not just diverse in terms of our composition, but in terms of where our faculty live, so to speak, across campus. Majority in School of Medicine, but we span actually 7 schools across UCI, including the School of Law, Pharm-Sci, Engineering, Bio-Sci, and our program in Public Health. And then I thought I should update a little bit as we're coming back together this fall, just on the kinds of things that we've been furiously working on through the center. As most of you know, and many people who attend these lectures were really supportive of our efforts and working with the California Institute of Regenerative Medicine and Americans for Cure to try and increase funding, again, up funding with the passage of Proposition 14 to fund stem cell research in California specifically, which was accomplished despite the pandemic in November of 2020. And that resulted in a period of time by the newly funded California Institute for Regenerative Medicine spent in terms of strategic planning and where to go over the next 5-year period. So we have been working feverishly in the center to try and take advantage of those funds for the campus, for our students, for the different kinds of progress that we can make. And so I have some really great progress to report on that. The first of those was a training grant for graduate students that we received actually in 2021. That again is focused on first gen and education of diverse students. Peter Donovan and Bogie Anderson, pictured here, really spearheaded that effort. But one thing that I really just am thankful for is what a fantastic collaborative team we have in the Stem Cell Research Center. And so many people contributed to this effort, all working together. And that resulted us in being the top ranked grant out of 17 proposals across the state. So we really killed it on this one. A second initiative that was brought forward by CERN was actually a new set of training grants focused on undergraduate education. And we just got news about 2 weeks ago that in fact we had the second ranked proposal in that category, and that is awarded to Brian Cummings, who's here tonight. And this is to support funding 2 years of complete support for undergraduate researchers to engage in training in the biological sciences, and really to offer new opportunities to people who might not be on a biological sciences track to begin with, but to receive training in clinical research relevant to stem cells and regenerative medicine, cell manufacturing relevant to stem cells and regenerative medicine, and have the opportunity to do research and be grounded in labs here. So hopefully you can see this slide. CIRM all along, as we've talked about through the years that we've been doing this program, is really focused on this idea of a translational pipeline. So moving out of basic science into clinical trials and in fact into clinical practice in the long run. We now have established that same training pipeline. So going all the way from undergraduate education to graduate education, postdoctorals, clinical fellows, and even we are implementing a program going forward that is going to be about training clinical professionals in how to do stem cell and regenerative medicine clinical trials. So towards that end, 2 initiatives that are already up in the air for CIRM involve reestablishing an alpha stem cell clinic network. We currently share an alpha stem cell clinic site partnering with UCLA, but we have an application pending right now. It's being reviewed this week for standing up an independent alpha stem cell ASCC alpha stem cell clinic here at UCI. And you can see in this graph what the trajectory of our stem cell and regenerative medicine clinical trials has been since 2019, with 60 projected clinical trials this year. So we feel like we're really in a strong place with this application. And our goal actually out of that is to form a network partnering with other Southern California institutions to really extend our reach into underrepresented communities and to all areas of California where there may be health disparities, to enable lots of people to be able to participate in this new medicine. The second effort that we're working on right now, but not the last one that's coming from CIRM funding, is about cell manufacturing. So we know CIRM has announced that there's going to be a good manufacturing practice, cell and gene therapy manufacturing set of centers that's established in a network across the state. The deadline for those RFAs for applications is going to be due in January, and we're already feverishly working on this. This is just a map to give you a sense of what UCI has already invested in that, building out a 7-room GMP facility that actually is going to be finished this November. And hopefully we'll be able to share more about that with you soon. The last initiative that's underway that, again, we're already preparing for is about shared research labs in core facilities. And I'm going to be excited to have the opportunity to talk more about that to you in the coming weeks or months. In the meantime, how can I help? You can sign up for our newsletter. You can visit us on our website. You can sign up for Facebook alerts. And really critically and importantly for us is, while research drives a lot about the US economy, certainly about the California economy for stem cells and regenerative medicine, it's not a huge source of philanthropy. So please do think of us in terms of making a gift, and the contact information is here. So with all of that said, just a couple of words on format, and I'd like to introduce our faculty speakers who are kind enough to join us for this evening. Because this is a hybrid format still, and you can see how, like frenzied it is to put that together, we're going to keep to our speaker presentations, and then, unlike in a dynamic interaction when everybody's here in person, hold questions for the end. You can put your questions up in the Q&A box at any time. We will be monitoring them. We'll answer them if we can, if there's a lot coming in, but we'll come back to them at the end to give a chance for both audience and those who aren't here tonight but are here virtually to be able to ask their questions and get them answered. So with that, our 2 speakers for this evening are Dr. Angela Fleischman and Dr. Matt Inlay. A couple of words about Matt and Angela. They're actually remarkably parallel. I was kind of chuckling when I put this together in terms of timing and location. Angela received her bachelor's at UCLA and did her MD PhD at Stanford. I know that they were both associated with the same lab, or Weissman's group there at Stanford. Matt received his bachelor's from Berkeley, did a PhD at UC San Diego, and then his postdoctoral fellowship at Stanford. We're really excited to have them both here on the faculty at UCI, and I'm totally going to put them both on the spot now just to break the ice a little bit, because Matt was pointing out to me, for example, that he has not even updated the growth in his family that has happened recently. So Angela, since you're going first, tell us something about yourself that you want the audience to know tonight. Why are you doing regenerative medicine research?
I'm doing regenerative medicine research because I want to do something important to help people. Maybe that's not so exciting, but that's why I do it. And I really love what I do because it allows me to, as a physician scientist, I can make connections with my patients over the years. And I can go back to the lab and take things that I learned from my patients and solve problems that will help people that I may never even meet in my entire life, and that's also very fulfilling to me.
That is an awesome answer. And tell us anything else you'd like to share about your lab, something personal. You have a totally putting you on the spot. I'm doing my best to be Peter Donovan, but he's much better at it than I am.
Now we only have 3 dogs, but we used to have 4 dogs. We have a house full of dogs.
That's a good thing to have. Matt, why are you doing stem cell and regenerative medicine research?
I think my reasons are very similar to Angela. I am a basic scientist by training, and while I enjoy basic science for its interest alone, I do feel like at this point in my life, I would like to do things that have a wider impact. And so anything I can do to better human health and do great science, that's what I'm going to do.
Fantastic. And do you want to update any of those personal details on the website so that?
Yes, my bio says I have a son and wife. I also have a daughter who is. I have a daughter who is now 7 and read my bio and corrected me for not including her.
They'll keep you on your
7
toes at that
years.
age. All right. Thank you guys for your patience. With that, Angela, can I invite you up?
You want to Zoom or
Yes. Okay.
Okay, well, thank you very much for this opportunity to speak today. The title of our presentation today is The Road Not Taken, Where Making Blood and Making Cancer Diverge. We're going to be focusing on blood stem cells, hematopoietic stem cells, and the immune system. So the hematopoietic stem cells or blood or bone marrow stem cells give rise to the entire blood and immune system. Hematopoietic stem cells are very powerful. The concept behind a bone marrow transplant leverages on the ability of theoretically a single hematopoietic stem cell to give rise to the entire immune system and blood system of a person. And this is why we can use bone marrow transplants to help people with certain diseases. There are different types of bone marrow transplants here. One type is called autologous, or you're taking the cells from yourself, freezing them down, and then giving them back to the same person. The other type is called allogeneic, and when we hear about transplants in the news, we're usually talking about allogeneic transplants. That's where hematopoietic stem cells are taken from one donor and then injected into a person with a disease. Although they're called bone marrow transplants, a lot of times the way that we get hematopoietic stem cells are not necessarily needing to go into the bone marrow to take the hematopoietic stem cells. We can give people hormones to make the stem cells come out into their blood and retrieve the cells from their blood. Now, each type of bone marrow transplant can be used for slightly different purposes. The autologous transplant, maybe a better way to think about it is high-dose chemotherapy with a stem cell rescue. This type of transplant is commonly used for diseases such as lymphoma or multiple myeloma, where we give the person very high-dose chemotherapy that without the rescue from their own stem cells would cause bone marrow failure.
Hey, Angela, hold up
Yes.
1 second. Online, your slides are not advancing.
Oh,
So I'm not sure why, but let's figure that out. Sometimes you drop- Can you see?
what is online?
We're seeing your presenter display, not the main display.
Oh, do you want to switch, or do we get, or do we get off of presenter view? Or
So you're saying that when we do that, you're seeing presenter view, or
Now,
is it good?
no, we're seeing the presenter view, not the other.
how about,
That's unfortunate. How about if we set up, do you want to just mirror?
yeah, we can mirror. Yeah, I
Or
don't need
is that
I
okay
don't
for
need the
you guys?
presenter view. That's fine.
This is online is good now.
Okay. And I guess we can deal with this here.
All right, good.
Okay. Sorry about that, everyone. Hopefully we're back in action now. Okay, so we were talking about the different diseases for which different types of transplants are performed. Allogeneic is when a person with commonly a leukemia will be given chemotherapy to put them into remission. And then to sort of, I guess you could say, clean things up, you take the bone marrow stem cells from another person and inject them into the person with the leukemia. I think it's important to note the utility of transplants in a leukemia. A person really does need to be in a remission before a transplant can be helpful for them. We really cannot give somebody a transplant with active leukemia, and I think that's a misconception probably in the lay media, that a transplant is just can cure. They do cure people from leukemias, but a person needs to be in a remission with chemotherapy before a transplant can actually help them. Oh well. So.
By going back and forth. There we go.
Okay, well, although hematopoietic stem cells are very powerful, they're also very vulnerable because we must have our stem cells throughout our lifetime. And if a stem cell acquires a mutation, this can be very long-lived mutation and can cause problems such as expansion, in this case, of a type of cell called a neutrophil. For example, a different mutation in a stem cell could cause expansion of a different type of cell, in this case, a plasma cell. And the type of cell that is expanded really defines the type of blood cancer that the person has. And humans have many stem cells on the order of 50 to 200,000 stem cells that must be maintained throughout their lifetime, from the time that they are in utero and a baby to adulthood. So in order to maintain stem cells, they do need to go under divisions to maintain their numbers. And with each division, the next slide was going to demonstrate that with each division, cells have a lot of DNA that they need to replicate, and it's inevitable that with the division, there will be mistakes in the DNA occasionally made, and these are called mutations. It's commonly known that certain types of stressors and harmful stressors such as chemotherapy, which are used to treat cancers, but can also have some harmful effects on normal cells. Smoking and chronic inflammation can lead to an increased risk of cancers, including blood cancers. So it had commonly been thought that these types of stressors create mutations in blood stem cells, and this goes on to then create a blood cancer. But recently, there's been new mechanisms that are important revealed for development of leukemias following stressors such as chemotherapy. In studies, when they have the ability to have blood from patients who subsequently develop leukemia, and they also have the blood before they ever got chemotherapy, they can find the cells that have the mutation that they thought caused the leukemia present in their blood even before they received the chemotherapy. So it changes the concept a little bit, that it's not necessarily the stressor that causes the mutation, it's that the stressor creates an environment that is conducive to the expansion of these already existing mutant cells. One way to think of it, on the slide, it's cut off, but before chemotherapy, I think half of them are, I think they're just cut off. That's fine, I can explain. Sorry about the technical difficulties. So I'm trying to display it in a picture that all of the smaller giraffes are the normal giraffes, and then you have this one mutant giraffe that is particularly tall. And when the leaves are very abundant and even low to the ground, the taller giraffe really is basically equal to the smaller giraffes. But after chemotherapy, when potentially the tree is very barren and the only leaves that are in existence anymore are the very tall leaves, now the very tall, quote, mutant giraffes now are the only ones who are able to get food. And so in this environment, now these pre-existing mutants that didn't previously have a selective advantage now are able to gain a selective advantage and expand. So in the lab, my lab is particularly interested in how a particular type of inflammation selects for a particular type of mutant blood stem cell that leads to a disease called myeloproliferative neoplasms. And we're very interested in myeloproliferative neoplasms because, interestingly, it runs in families, and also coinciding with the tendency to get a myeloproliferative neoplasm is also autoimmune and other inflammatory conditions. So I'm making an attempt to explain this, this mouse experiment here. We have mice that have been transplanted with both mutants, the pink cells, and the normal cells, the blue cells, so they're in a mix. And we can detect the mutant versus the wild type by cell surface markers on these cell surface. So we can follow in the mouse's blood what cells are mutant and what cells are normal. And so as you can see, we start an exposure, which is a specific type of inflammation, at about 50 days after the mice are transplanted. And we follow the mice that get the inflammation and just the saline, the control, over time. And as you can see in the purple with the inflammation, the mutant cells expand more at the expense of the wild types or the normal cells only in the inflammation scenario, whereas in the saline scenario, the mutant cells actually decrease over time. And at the end of the experiment, we also measure spleen size, which in this disease, patients can get an enlarged spleen, and that is a measure of their disease burden. You can see in the inflammation group, the mice had spleens were more enlarged as compared to the saline group. Now, based on my laboratory's research, we wanted to take things into the patient realm to try to identify approaches, ideally low risk and low cost approaches to reduce inflammation in blood cancers. We thought about 3 different aspects: medication, microenvironment, meaning the gut microbiome, and lifestyle changes such as diet. For medication, we were very interested in the antioxidant N-acetylcysteine, which is currently used widely as a supplement and is also used for Tylenol overdose. We tested the utility of N-acetylcysteine in our mouse myeloproliferative neoplasm model. We were originally not necessarily wanting to look at blood clotting in this model, which is a key characteristic feature of the model. We were more interested in inflammation. However, the effect on N-acetylcysteine in preventing lethal blood clots was quite impressive in this mouse model. And so we wanted to further investigate how this supplement could potentially reduce blood clots in this model, in addition to inflammation and as well as protecting blood stem cells. In this experiment, we were specifically asking whether N-acetylcysteine can protect normal blood stem cells from chronic inflammation. In mice, if you expose mice to chronic inflammation and then take out their blood stem cells, they are exhausted, meaning they do very poorly. Like in the red in the middle, that those cells are coming from the inflamed mouse, and they're doing less well than than the cells from the mouse had just been given PBS or the saline. But then in the right, which we gave the mice inflammation plus put N-acetylcysteine in their water, and that totally reverses the negative effects of the inflammation on the blood stem cells or the hematopoietic stem cells. So now we're moving on to a clinical trial in progress here at UC Irvine, which our primary objective for this study is to identify the optimal dose of N-acetylcysteine to give to patients with the chronic blood cancer myeloproliferative neoplasms. And currently we're testing 3 different doses of NAC, and our goal is to identify the dose that we can see an impact, but is tolerable to take for patients. Next, we wanted to look at the gut microbiome. The gut microbiome is an emerging topic in health and disease, but the microbiome in blood cancers, and in particular in myeloproliferative neoplasms, is really quite unexplored. So in this study, we took 25 MPN or myeloproliferative neoplasm patients and 25 healthy controls, took their stool samples, as well as blood from a subset of people to measure inflammatory proteins in their blood, and compared the gut microbiome of MPN patients to healthy controls. In this, when we look broadly at richness of the microbiome, which means the number of species in the gut, as well as the evenness, meaning how well each species is represented, we didn't identify a significant difference between MPN patients and normal controls. However, when we looked more closely, we found that there was an underrepresentation of a specific bacteria called Phacocolobacterium in MPN patients. And interestingly, this bacterium has been previously demonstrated to be protective from inflammation. So the fact that MPN patients have less of this bacteria, which could be protective from inflammation, one could surmise that this may play a role in why there is high inflammation in this patient population. And then we also wanted to look at, this is a schematic of each of the colored bars. The different colors in the bars is different types of bacteria in each person's gut microbiome. Again, sorry about the slide, but what I want to communicate is that each person's microbiome is very unique to themselves. So each person is much more similar to themselves than any other person. So it's very, very difficult to identify particular things such as disease or interventions that change significantly the microbiome, because your microbiome is always going to be your own microbiome. However, from this, we found that 1.7% of the between cohort variance in the microbiome can be explained by a diagnosis of a myeloproliferative neoplasm, which, when you put into context, a very large intervention such as a colon surgery can explain about 5%. So 1.7 is quite a significant difference. Next, we also wanted to look at whether we could reduce inflammation in blood cancers through diet. Because in other chronic inflammatory diseases, such as cardiovascular disease, a Mediterranean diet has been found to reduce biomarkers of inflammation as well as improve outcomes. So we thought that maybe myeloproliferative neoplasm patients, which also have chronic inflammation, could benefit from a dietary intervention such as a Mediterranean diet. So the properties of the Mediterranean diet that make it, quote, low inflammatory: it's very rich in fiber, which is anti-inflammatory, is rich in antioxidant compounds such as resveratrol, which is seen in red wine, and also is rich in monounsaturated fats, which comes from most of the fat in the Mediterranean diet comes from olive oil. So we thought our hypothesis, or our overarching goal is we think that by following a Mediterranean diet, this might reduce inflammatory proteins in an MPN patient's body, and thus improve their symptom burden, and maybe could also blunt their disease trajectory. This is a picture of, sorry again about the slides, a picture, a schematic of our first diet study. The schematic is supposed to be in the right there. What we did was we took 30 participants. They all had myeloproliferative neoplasm. We split them into 2 diet groups, Mediterranean versus just a standard USDA guidelines for American group. We gave them in-person dietitian counseling, and we also gave them weekly colorful PDF curriculum. We drew biological samples, including a complete metabolic panel, complete blood counts, high sensitivity CRP, lipids, and plasma cytokines, which are the plasma inflammatory biomarkers. And what we learned from the first study, our primary objective here was simply to see if we give MPN patients dietitian counseling, can they change their diet towards a Mediterranean eating pattern? And that's in the pink. We wanted to get over 80% of our patients on the Mediterranean diet, achieving a high Mediterranean diet score, which we defined as 8 on a 14-point scale. And we did achieve 80%, over 80% of the people on this diet arm did change their diet to be much more adherent to a Mediterranean diet. And on the right are what's called waterfall plots, where each bar is a patient. And in this disease, symptoms are very important, and they are measured with an objective scoring tool called the MPN Total Symptom Score. And many clinical trials in this disease, an important endpoint is reduction in this score. So we also wanted to look at whether a Mediterranean diet could reduce the symptom score in this disease, and that's the Mediterranean diet is in the pink. And as you can see, many of the bars are going down in the pink arm. However, in the USDA arm, many patients also did enjoy a reduction in their symptom score. This is not unexpected, given that realistically, if anybody improves their diet, they can expect to feel better, but it does appear that patients in the Mediterranean diet did enjoy a more impactful reduction in their symptom burden. So other exploratory biological correlates that we measured that I don't necessarily have time to get into were blood counts, cholesterol, CRP, inflammatory proteins in the blood, and the gut microbiome. But just these are the plasma cytokines or the inflammatory proteins in the blood. And this was what's called an exploratory endpoint. We simply wanted to observe. With the small numbers of patients that we had, we didn't necessarily expect to see a difference, but we just observed. And we did not see any significant changes in their inflammatory proteins in their blood. However, a much larger sample size is required to really expect to see changes. And we also looked at the gut microbiome in these patients. And as you can see, each person is very unique and much different than each other. We did not detect any discernible changes in either of the group's gut microbiome throughout the 16-week study, which is not necessarily unexpected as well. So our next steps with the diet intervention in MPN, as I mentioned that our first study had a very small number of patients. We'd like to move on now to a much larger group, which is what we say powered to detect changes in symptom burden. To reach a much larger patient population, we're going to do all of our dietitian counseling online through Zoom. And different than our previous studies, we're going to include a standard of care arm, basically a no diet intervention, because we feel that any diet intervention can be helpful for a person. So in order to really tease out the impact of a diet intervention, we're going to have just an observation arm. And we're going to make a personalized Mediterranean diet, which includes fermented foods to maximize changes in the microbiome. And last but not least, the acknowledgements for the mouse studies. previous grad students, Betty Lai and Bree Hoover, as well as a current postdoc, Raja Ramanathan, and a current grad student, Jeanette Sullivan, for the microbiome was done by a visiting scholar, Kenza El-Alawi, who is currently back in Belgium and actually is from an MPN family. So that was very nice for me to be able to work with somebody who was personally touched by myeloproliferative neoplasm and allow her to participate in research in a disease that affects her family. And then the diet intervention was driven by a previous grad student in my lab, Laura Mendez, with help from undergrads in my lab, and was a collaboration with another investigator here at UC Irvine, Andy Odegaard. Thank you very much.
So again, we're going to go straight through to our second talk and then take questions at the end. So Matt, if you would be so kind.
All right. Did it stop sharing? No, it's. In person and via Zoom. Hi, everyone. Seems like the volume keeps going out. I'm going to talk to you today about my research looking at the immune system and how we can balance activity and tolerance after a bone marrow transplantation. So Dr. Fleischman already gave a beautiful introduction on the hematopoietic system, so I'm only going to touch on a few quick points in the interest of time. So our blood system comprises not only red blood cells and platelets, but also all of the cells of the immune system, and that includes our innate immune cells, which are quick-acting but very generic response, and our adaptive immune system, which has lymphocytes such as B cells and T cells that can respond in a very specific and highly adaptable manner to infections. All of these cell types are produced by the hematopoietic stem cell, also known as the blood-forming stem cell or the bone marrow stem cell. And I'm going to be calling them HSCs throughout my talk. They have three properties that make them very useful in regenerative medicine. Those include their ability to self-renew, so they can make identical copies of themselves. That's often indicated by this circular arrow here. This is a property that all stem cells share. They are multipotent, meaning they can make more cell types. So in this case, for the HSCs, they can make all of the cells of the blood system. And then lastly, they are engraftable. So that's a little bit of a trickier concept, but it basically means if you introduce these cells into a host intravenously, they will circulate, they will find their way to their microenvironment, their stem cell niche, as we often call it, which is in the bone marrow. So they will find their way into the bone marrow, they will enter and seed there, and thereby, once there, produce more of themselves and produce all the cells of the blood lineage. So the combination of these three properties makes them a very valuable tool in regenerative medicine. As Dr. Fleischman mentioned earlier, you can use these in a transplantation system to basically replace a patient with a diseased blood system to replace it with that of a healthy donor. So this is a very simplified cartoon of the process. You can take a patient with a blood disease, you have to remove their diseased blood system, and then you can replace it by extracting either bone marrow or nowadays mobilized blood from a healthy donor, processing their cells, and then transplanting them into the patient. And if everything works, you will completely heal that patient of their blood disease. So as you can imagine, this treatment, this strategy can be used to treat almost any blood disease. It's mostly used to treat blood cancers, but you can also potentially use it to treat genetic blood disorders like severe combined immunodeficiency and sickle cell disease, and even potentially things like autoimmunity. So if you can imagine using this process to basically reset your immune system. The problem is there are many risks involved with it. So these are just a few of the complications that can occur. You can imagine while you are recovering from this procedure, you're very vulnerable to opportunistic infections. You can have graft failure or rejection, or the condition that I'm going to be talking about today, which is called graft versus host disease. And this is a complication that stems from the fact that the donor graft contains not only the stem cells, but often contains immune cells either from the blood or the bone marrow. And these immune cells can view the host, can view the patient as a foreign entity and begin to systemically attack it. So while the donor stem cells are engrafting into the bone marrow and producing more blood lineages, the donor immune cells are attacking the patient at many different organs, including the skin, liver, and gut. So this can happen in about 30 to 60% of allogeneic transplant patients, so patients receiving their cells from a donor. It involves many symptoms, including issues with the skin, the gut, the liver, basically systemic inflammation. And you can treat it using very powerful immunosuppressants such as glucocorticoids, which is also going to be the focus of my talk today. So because of the risk of GVHD, this is why you often hear this phrase matching, while the donor and the patient need to be matched in order to prevent this from happening. So what do I mean by matching? So I have to do a little bit of immunology lecture right now, so bear with me if this seems a little technical. We have T cells. These are immune cells that circulate in our blood. They're responsible for detecting pathogens that hide inside of cells. So here in this case, we have a healthy cell. It produces proteins kind of indicated in green. And what T cells do is they scan these cells to look for signs of infection. The way they do that is that there's a system in place where every cell displays a portion of its interior proteins on the surface using a complex called major histocompatibility complex, or in humans, we call this HLA. And so there are tens of thousands of HLA molecules on the surface of every cell, and each one of them is displaying a piece of a protein that's in the interior. So there are tens of thousands of interior proteins being displayed on the surface, and T cells will scan these HLAs to see if there's any foreign proteins present. So in this case, this is a self protein in a healthy cell. So the T cells are educated not to respond to these. So there will be no recognition in this case and no response. Now, in the case of a viral infection, here in this example, you have a virus in red that has infected the cell. And so there are viral proteins inside of the cell. These are displayed by the HLA molecules. And now the T cell can recognize this and it will respond to this. And it does it by attacking the cell or secreting factors that induce inflammation or encourage other immune cells to attack these cells. So this is how it's supposed to work. But in GVHD, what happens is the T cell has not been educated on the same background, the same HLA as the host. And so a subset of these T cells will actually think that the HLA itself is a foreign molecule. It will recognize it and it will do the same attack as though this was an HLA displaying a foreign molecule. So this is why there must be careful matching, because the only way to prevent this from happening is if the donor T cells are educated on the same HLA molecules as the host. So when you get, when people are typed to see if they're a match, they're screening their HLA alleles to see if they have the same genetic sequence. So many of you probably are familiar with the bone marrow registry. Nowadays, you can just contribute either a saliva sample or even just a cheek swab to be screened to have your HLAs typed. And patients who need a bone marrow transplant that need a match, they can find either from a relative, about 30% of the time they can get one from a relative, but about 70% of the cases they need a match from an unrelated donor. And this registry can offer the potential to find a match. The odds are still fairly low. I think it's about 1 in 10,000. And for certain ethnic groups, there's abundant donors on this bone marrow registry, so they can find a match easily. But then there's other ethnic groups that have a much fewer people contributing to this registry, and it's a lot harder for them to find a match. So one alternative is you can use haploidentical HLAs or half matches. So this is where some of your HLAs are matched between donor and host, and some of them are not. So all parents are half matches to their children and vice versa, and about half of siblings are half matches to each other. So the problem with this is there's this high risk of GVHD because there are HLAs that are not identical, and there's a risk of your T cells responding to them. So one of the goals of my lab is to find new ways to improve the safety and efficacy of bone marrow transplantation. And one of the ways we are doing that is looking for ways to either reduce or prevent GVHD, and that will hopefully allow an expansion of the donor pool to include more of these haploidentical matches. So my work involves a class of drugs that we call glucocorticoids that are steroid hormones. Some of them are produced naturally, like cortisol. Others are synthetic. They act on a very wide range of pathways and systems in our bodies. They control metabolism. They can control homeostasis, but they're also involved in the immune system. And in the immune system, they're mostly known for their properties in anti-inflammation and suppression of the immune response. So there's a lot of synthetic glucocorticoids out there you can buy or use, ranging from simple ones like cortisone for anti-itch creams, Flonase for reducing allergies. But then there are stronger ones like dexamethasone, which was shown to help reduce COVID-19 symptoms, and then even stronger prednisone, which has anti-cancer properties, as well as a drug that you might take if you start to develop GVHD symptoms. And so this brings me sort of to my first analogy here, is that the immune system has to be very delicately balanced, because on the one side, if you have too much activity, you can lead to overactivation and chronic inflammation. And then on the other side, you need to balance it because you still need an effective immune response. So if you weigh too heavily on this side, you can end up with suppression or an ineffective immune response. And so the advantage of glucocorticoids is it's a very easy way to sort of tilt the scale away from overactivity and towards immune suppression. But too much and you can have an ineffective response. So the first experiment that I want to show you was done by a former grad student, Erica Verratti, and she wanted to see the effects of glucocorticoids on immune cells. We tested a variety of glucocorticoids. The one we settled on was called fluticasone propionate, better known as the anti-allergy drug Flonase. She harvested tissues from mice, their bone marrow and their spleen. She cultured them in either Flonase or what we call a vehicle control. These are just, vehicle is just what Flonase is dissolved in. Oops. And then she measured the viability of the cells after culture. And when she looked at the T cells, so these are the cells that are primarily involved in GVHD, she found a pretty surprising decrease in the viability of the cells after 16 hours in culture. So this is a bar graph of the percentage of live T cells either after vehicle treatment or Flonase treatment. And you can see the viability drops pretty dramatically after 16 hours. So the Flonase is basically inducing death of these T cells. So we thought perhaps we could use this as a strategy to combat GVHD. Now, before I proceed, I should mention there have been clinical trials that have used these drugs as a way to prevent GVHD by giving them to patients before they even develop symptoms. But those trials failed to show any benefit, likely due to the fact that these patients had just undergone a bone marrow transplant. So they were already immune suppressed as it was. And so this is giving them a powerful immunosuppressant when they're already vulnerable to infection, probably countered any benefit that it might have had. So you can't really give this to the patients before they develop GVHD to show benefit. So we came up with a different strategy, which is rather than treat the patients, why don't we treat the cells first? So our idea was we could take our donor cells and pre-treat them with different glucocorticoids prior to transplantation. So rather than give the patients the drugs, we give it to the cells. So this is a classic GVHD mouse model where you take one strain of mice, harvest the spleen, the bone marrow, and transplant it into a second strain of mice that is a mismatch. And these mice will very rapidly develop GVHD within 7 to 10 days. So in our strategy, we basically, rather than directly transplant these cells, we first culture them in either Flonase or vehicle, and then we do the transplant. And when we do that and look at the mice later, I have to, this doesn't play unless I escape. Okay. All right. Okay, and when we look at these mice, this is 28 days after the transplant. You can see one mouse looks relatively healthy. The other one is suffering from fairly severe GVHD symptoms. And the healthy mouse was given the cells pre-treated in Flonase, whereas the mouse suffering from GVHD was given the cells pre-treated in the vehicle. So a pretty dramatic difference in this example. Okay, and so we could score these mice. So we're based on their symptoms. So there's sort of 5 classic external GVHD symptoms. You give 1 point for each one, and you can give them what we call a clinical score. And we can track this over several weeks. And you can see here that the clinical scores increase over time. For the mice given the vehicle-treated cells, they continue to rise until they reach sort of the maximum score of 5. Whereas the mice given the Flonase-treated cells kind of peak at around a clinical score of 2. So we're already showing here that our pre-treatment strategy has a reduction in the severity of GVHD. So what about survival? So this is what we call a survival curve, where you are tracking what % of the mice are still alive after so many days after their transplant. And you can see in the vehicle condition, most of the mice are succumbing to GVHD. All but one died within 10 days, and the one that survived to 28 days was the mouse that you saw in that video. Meanwhile, the mice given Flonase-treated cells, some of them did succumb to GVHD, but many of them survived, and about half of them survived to the end of our experiment window, which is about 120 days, and showing no additional signs of GVHD. So we saw greatly improved survival. And remember, we didn't actually give any drugs to the mice, right? The only difference between these mice was done prior to the transplantation. So nothing was injected into these mice, so no immunosuppressants were given to these mice. So our next question was, why does this work? All right. So this is data I showed you previously. This is looking at the viability of the T cells after our culture. So what do the T cells look like in the mice? Are they similarly depleted? And the answer is no. They start to catch up pretty quickly. So this is 7 days after transplantation, looking in the spleen. This is the percentage of immune cells in the spleen that are T cells, and you can see they're roughly equal between the two groups. So even though we are initially wiping out a large fraction of the T cells, they quickly recover in our recipient mice. And these mice have almost very similar numbers of T cells. So it can't be simply that we're wiping out all of the T cells that are causing GVHD. This is a graph looking at the percentage of T cells that are activated. So we look at markers of activation to see if we can detect the difference between the two groups. And you can see here that about 17% of the T cells given to the vehicle that were vehicle treated are activated, whereas only about half of that were activated in the Flonase group. So we are seeing a decrease in activation, though not as dramatic as you might expect, but still we're seeing decreased T cell activation. So the last thing we wanted to look at that I'm going to show you today is looking at a rare population of T cells. So most of what I've been talking to you about so far has been what we call conventional T cells. So these are immunogenic. These are the ones that cause GVHD. But there is a rare population of cells that we call regulatory T cells that have the opposite function. They're immunosuppressive and they're tolerogenic. So in this example, this is sort of mimicking that GVHD condition where you have the donor conventional T cell recognizing the host HLA and attacking it. What the regulatory T cell does, or Treg, when it recognizes its target, it actually releases an inhibitory signal to prevent any neighboring cells from acting upon their targets. So it's suppressing the immune response. And even though regulatory T cells are very rare, they're only about 4% of T cell population, they're very important in preventing things like autoimmunity. So if you wipe out the Tregs in a mouse, they will get autoimmunity very quickly. So even that 4% is very important for preventing that. So if you think about the scale analogy again, even within the T cell populations, there has to be a balance. So too many conventional T cells or too strong a conventional T cell response, you get overactivity and GVHD. But if you have Tregs, you can get tolerance. And if you have too many Tregs, you might get an ineffective immune response. So we decided to look at Tregs. This is now work done by a current grad student, Angel Ayala. This is data you've seen 3 times now, where we're looking at the death of conventional T cells after Flonase culture. But look at the Treg population. They are basically unaffected by the Flonase culture. And this leads to an increase in their relative frequency. So where in the vehicle group, they're only about 4% of the T cell population. After Flonase treatment, they expand to about 12%. So about a 3-fold increase and a change from the ratio of Tregs to conventional T cells from about 1 in 25 to about 1 in 8. And this maintains in our recipient mice. So if we look in the spleen 7 days after transplant, we see a similar increase in Tregs. So we're doing a lot of other work that I don't have time to show you today. So to sort of wrap up what we think is going on, we think in a mismatched transplant, what you're seeing is overactivity by the conventional T cells. They're responding to the mismatch and causing GVHD, and the Treg response is not strong enough to counter that. But by altering the fraction of Tregs in our transplants, in our graft, we're shifting the balance and creating a more tolerogenic environment. So just by changing that ratio of Tregs to conventional T cells, we're creating this sort of tolerogenic environment in the host for the donor T cells. And so just to show you some of the directions we're going in, we still want to understand the mechanism further. So we're looking at a variety of things, one of which is trying to understand why the Tregs are unaffected by these different glucocorticoids. We can consistently get about 50% survival in our GVHD model. We want to see if we can get that even higher. So if we augment these ratios further, can we get even better survival than what we're seeing? And then most importantly, can we get this system to work in human cells? And so our hope is that if this works in humans as well, or we can get this to work in humans as well as we can get it to work in mice, we can modify the existing treatment strategy to include a step where you do a glucocorticoid pre-treatment prior to the transplantation to help condition the donor cells to be more tolerizing to the host, and thereby you can heal the patient without developing GVHD. And so just to thank some of the people in my lab, this work was done primarily by Erica Verratti, who is a former grad student. Her work was continued by a newer grad student, Angel Ayala. And these are just many of the people in my lab, including my lab manager, Kewen, who also did a lot of the human work on this. And then I also want to thank our flow cytometry core, Vanessa and Pauline, for helping us do most of the analyses you saw on these slides. And then lastly, most of the funding for these projects was done by a donation. So the Oxnard Foundation gave us a small grant in order to conduct these studies. And without that support, we would never have been able to pursue this project. And so with that, I will end and we can now take some questions. Thank you, guys.
I can't believe you guys landed that on. Thanks.
Yep. Is that going? Yeah. Apologies for the technical difficulties, but welcome to the new Zoom. They always release a release like right before we're doing a lecture, I swear to God. So really quick, maybe just to kick us off, we have a couple of questions that have come in on the chat, and I would encourage anyone who's with us online to please post your questions in the Q&A. So I'm going to ask you guys a question in general and then kick it out to this audience here, and then we'll start picking some up from online. My question to you, Matt and Angela, particularly because of, you know, the fact that both of you spent time in Irv Weissman's lab, who was so foundational in terms of developing this type of therapy, right? The idea that you could do hematopoietic stem cell transplants, isolate them, do something with them. And you guys give wonderful talks, but I don't know that that full background was kind of conveyed and came through. So here's my question to you. Can you just give us a little bit of context? I'm trying to think of a good way to phrase this for why hematopoietic stem cells are so important in the stem cell field. Like, what does that represent in the context of regenerative medicine? Because really they're our first stem cell treatment, right? So can you spend a couple of minutes and just talk about that?
Well, I think Dr. Fleischman would be better
Or one
suited
of you.
than I would, but while she's getting her mic, I'll just say this has been a procedure performed since at least 50 to 60 years now. And so it is arguably the most thoroughly studied stem cell therapy out there.
And I think that, I guess you are right. We probably took for granted all of the work that was required to sort of dissect each cell in the bone marrow with different antibodies and identification of what cell surface markers define a hematopoietic stem cell. I think that because the bone marrow and blood is liquid, you know, you can mush it up and it's each cell, honestly, each cell is its own organ as opposed to solid organs where it has a structure and you have to, it's difficult to get a slurry of cells. And probably flow cytometry also really, hematopoietic cells are very conducive to flow cytometry, and that really allowed to identify and select for particular types of cells and to do experiments.
So is it fair to say that this is like the first example of a regenerative medicine treatment?
I would say yes. Yeah.
Because I think that context is really important to convey, right? Because here we are 40, 50 years later, right? And we're on the cusp of thinking about so many different types of treatments. But this is like in a lot of ways where it started with stem cells.
Correct. And I think we're in the hematopoietic stem cell field, you know, bone marrow transplants have been around for so long, and just, you know, transplanting bone marrow from one person to another, I don't want to say is commonplace, but then that allows in the hematopoietic field for us to go one step further, that we can, further become more sophisticated in how we utilize hematopoietic stem cells for health.
Right. I'm going to steal that from you.
And maybe you guys could share that microphone. Do we have any burning questions? Yep. Hold on, Anita.
I have a question. Would you please clarify for me? So, did you treat the T cells with the HSCs? So that was the mixture of the HSCs and the T cells.
In some experiments, we did, and
So my
go ahead.
second question, did you see any impact on the survival of the HSCs?
Yes, so actually, it actually has a very dramatic impact benefiting the HSCs. So yeah, so this is actually how we got into this, because we read a paper where a team used specifically Flonase to induce the upregulation of a molecule called CXCR4 on the surface of HSCs, human cord blood HSCs, and they saw improvements in cell migration ability and homing. So we already knew from the beginning that these glucocorticoids were not going to hurt HSC engraftment, and in fact could provide a benefit. So our data is showing that not only do you see a benefit there, but you're also seeing a benefit potentially in eliminating or reducing GVHD. Great question, though.
Yeah.
Yeah. So since the Tregs go up, do you think that those animals would have a greater chance of getting tumors later?
The potential is definitely there. So one of our concerns is we still need T cells, right? We don't want to create an environment where there is no T cell function at all. And so the trick here is, how do we figure out the right balance? How do we prevent GVHD while allowing normal T cell function to work? And in fact, there's a very important response of T cells called graft versus tumor or graft versus leukemia, or GVL, where you actually want the T cells to act against the tumor. So the trick is, how do we steer them in that direction and away from GVHD? And so that's one of our future goals, is to be looking specifically at some of the properties of these T cells after this treatment to make sure they still have some functionality.
I'm going to jump and try and take a couple of questions from online. One that came in early on, which is definitely to you, Angela, is what is the difference between a myeloproliferative neoplasm and a myeloid malignancy arising from the same progenitor cells, like acute myeloid leukemia?
Okay, so a myeloproliferative neoplasm is a myeloid disease. So in blood cancers, the hematopoietic stem cell first makes the decision of to become a myeloid cell or a lymphoid cell. And many of the leukemias, chronic leukemias and acute leukemias are first designated as myeloid or lymphoid. And then depending on the specific type of mutation that's acquired, as well as the type of cells that are expanded, can define what type of disease the person has. So a myeloproliferative neoplasm is a myeloid malignancy. It can turn into a leukemia. The most common mutation seen in a myeloproliferative neoplasm is a JAK2 mutation. Interesting, some leukemias, acute leukemias, can also have a JAK2 mutation where the person didn't have an MPN to begin with. So I hope I answered the question, but it's depending on the clinical presentation of the person, sort of defines what the person is diagnosed with, and people can sometimes change from one diagnosis to the other as their disease progresses.
Great. So we'll keep an eye. We may have a follow-up to that. I'm going to turn to Matt, and then we'll come back to the audience for one. Matt, a question that we have online is, lots of people take Flonase for seasonal allergies, for all kinds of reasons.
I'm taking it right now.
Yes, and so am I. And you described really well this the consequence of cells being exposed, of T cells being exposed to Flonase. You're depleting those good T cells, right? And maybe increasing regulatory T cells. So if you're on Flonase, the question is, does it matter in terms of your susceptibility to viruses then? Because are you depleting the T cells that would normally be fighting that off?
No, I don't think in humans it has the same effect. So we've been trying to replicate what we've observing in mouse cells and human cells, and it's very different. So the human cells, it has a much more mild reaction. So we're looking at other more powerful glucocorticoids to see if we can create that effect, but definitely you don't have to worry about your T cells if you're on Flonase like you and I are. So.
Probably half the room. So one more quick one, and then I promise I'll go back to you guys. And because I think it is directed at you, and the question is how long until clinical trials? So Angela, you talked already about some of your work that's led into clinical trials. So I interpret this question as being, is it viable to be able to incorporate what you're studying in the lab into mechanisms to be able to block or stop the development of graft versus host disease in a clinical trial going forward?
Yeah, so I mean, I'm a basic scientist, so I may not be answering this correctly, but my impression is that we need to do two things. We need to understand the mechanism by which it works, and then we need to be able to find that mechanism by treating human cells. So we don't have the luxury of, doing the mouse experiments with humans. We can't we can't just inject these cells in humans and see what happens. So we need to figure out what is happening to these cells in a dish, and we can use that same detection method to see if we can reproduce that with human cells in a dish. So until we know exactly why it's helping in the mouse system, I don't think we can start thinking about clinical trials in people yet. So, but it's tempting. It's definitely tempting.
Angela's going to argue with you.
And I think that the time frame from initial discovery in the lab to actually reaching people in the common scenario probably takes decades. I mean, I could see that the next step would be doing some safety studies in humans, which would, we first have to make sure that our approach is safe. So, you do make sure in the lab, you're comfortable, it would be safe to test in humans. You test it in a small number of humans to look at safety. And then you look and see, do you see a signal that it's having the effect that you're hoping? And then you can move to a much larger study. And then after that, bring it for FDA approval, but that would take decades. Or I don't know, maybe not decades, but a long time.
Arguments, because I'm making a face over here a little bit. Or the purpose of argument here, you made a case, Matt, that this is a really serious complication to trying to treat what is already going to be a fatal disease. So you have people out there who I would imagine could be sitting in their living rooms, or if one of us here was in this situation, and we might say, really, is that the right way to be looking at the risk benefit ratio? I mean, you're a clinician, right? So just pushing on you, right? What is
well, I guess that would be who would be phase 1 studies, or people who are, I know it's desperate and really have no other options. So in them, they're able to take the risk of an unknown, of something that is simply being investigated to see the safety. I mean, for transplants, because the utility of Flonase appears to be
totally
prior to transplant, you don't know whether the person's going to develop graft versus host disease, because the intervention is before the transplant. So you have no knowledge of whether that person was going to be destined to develop terminal graft versus host disease or not. So I maybe the situation of having somebody very desperate would not necessarily fit towards Matt's situation, because it's prior to their transplant.
If you're up to 60%, right, as high as 60% graft versus host, that's pretty significant as a consequence.
Yeah, I mean, there are other strategies out there to try to mitigate it as well. So I think we would have to show that we were better than those other strategies.
Sure.
So,
Audience, what do you guys think? Somebody that's not Anita. We have students here. Come on. All right, good. Hold on one second.
I have quick question for Matt regarding what happens when you had a pretreatment, and was it just a matter of T cells being either killed or staying alive, or was there some damage also to T cells in the process? So some went completely 100% healthy and some might have had some damage, maybe even had to recover somehow? Or was it just nice and clear, yay or nay?
So within the 16-hour culture, most of the T cells were either undergoing apoptosis, which is called programmed cell death. I don't think that is recoverable. So the cells that have initiated apoptosis will complete it. And then most of the cells were actually already past that stage and completely dead. So I would say in 16 hours, most of the cells that we were saying were not alive were definitely dead. And they're not going to recover. So what you're seeing when we transplant that the T cells are recovering, that's from the rare population that did survive the culture. So.
So I was looking at the one out, and I'll just walk back to you right now. What happens to the B cells after glucocorticoid?
So all of the lymphocytes, so that includes all of basically the entire adaptive immune system, the B cells, the T cells, even a population called natural killer cells, they all suffer about the same level. Some more than others, but they all basically are being killed by this drug. And the innate immune system is completely fine.
A question for Dr. Fleischman regarding, I read some study very interesting about the Mediterranean diet. For different countries in Europe, you have different results for the respective populations and how efficient or efficacy of the Mediterranean diet. How do you protect yourself for that same phenomena when you administer it to future group that will be larger?
So
And-
is what you're saying is the Mediterranean diet means something different in
No.
Spain or
No, the
the
same thing.
genetic background?
The same thing, for example, in Poland as in Italy, but you start to see difference for the respective demographics in the results. Still beneficial, but for some better than for others. So there might be some genetic effect that comes into play over large enough of a population. And question is, can the same phenomena occur in North America?
So with regard to, with regard to how different interventions may affect different people based on their genetic makeup, yes, that is a situation that in in clinical trials, other than at the present time self-reported racial background, we don't necessarily do more than that in terms of like 23andMe typing them and things like that. But we try to put people, stratify people in things that we believe would be important for the outcome, like age, their sex, what treatment they're on, what stage of disease they have, so that there's equal number of people in both groups. I understand I'm not quite answering your question here, but we try to normalize the groups to the best of our ability for things that we believe may impact how they respond to the diet. But genetics is not necessarily one of them.
But presumably that'll be
could,
randomized
yes,
in your group.
yes.
Right. Yeah. In a large enough group. Any other questions from our audience tonight? Otherwise, we're coming up on 8:30. Good to go. All right. Thank you guys very much. Thank you for your patience and your tenacity with our AV issues tonight. Thanks to the audience for joining us here and virtually, and have a good evening.
Thank you.